So now I will be starting with competency 3.3. We'll learn today nerve injury and how these are classified. Then we'll be learning degeneration. When a nerve fiber is injured, then it undergoes various pathological changes along the nerve fiber. So what are these pathological changes, how it takes place? What are the different steps? So that is mechanism of degeneration of peripheral nerves. This is also called Wallerian Degeneration. Wallerian Degeneration. Then again we'll learn the mechanism of regeneration of peripheral nerves. So when these are injured, there will be degenerative changes, and after that, depending on certain criteria, they will undergo regeneration, the revival of the nerves. We know the nerves cannot be reproduced because they are unable to divide. So only one way is their degeneration. When they will be injured, they can be regenerated. But it depends on certain conditions. We will learn these conditions. Shamit, with your copies. Come with your copies. Sit there. So, in our last class, we have seen that every axon is having an axolemma, and this is again covered by neurolemma. And if this neurolemma is again covered by endoneurium. So this is endoneurium. Myelin sheath, here you will get neurolemma, endoneurium, and all these fibers make a bundle. These bundles are called fasciculus. Now fasciculus is having another cylindrical membrane, perineurium. Now lot of fasciculi will be bundled together within another membrane, membranous structures, connective tissue membranous structure. This is called epineuria. So you are getting a nerve trunk. So within this trunk, what you will be getting? Numbers of fasciculi. And within this each fasciculus, what you will be getting? Numbers of nerve fibers. So nerve fibers has got a covering. Bundles are also having a covering. And all the bundles together, they are forming trunk, and this trunk is also having covering. Okay, so this you should understand. So this, if you take a cross section, this will look like this. Now, nerve injury. And injury to a nerve can happen by various means. One is you simply press for prolonged time. Then what will happen? This part you will feel, the later part, the proximal distal part, you will feel there is lack of sensations. You are losing sensations. You experience it so many times. So this is the first degree injury. If this is prolonged over a period, then the terminal part of the nerves can be damaged. So pressure is one thing. How pressure is when you are pressure or by there is obstructed. Suppose here it is obstructed. So there will be lack of circulation. Since there will be lack of circulation, we call it, when there is lack of circulation, we call it ischemia. In heart, we know there is a disease called ischemia, ischemic heart disease. What is there? On the wall of the heart, they are having their own circulatory vessels, and these vessels are supplying oxygen and nutrients to the heart muscles. Now, if these vessels are narrowed or obstructed, the area it is supplying will not get sufficient oxygen, nutrients. They will feel, these cells will feel there will be ischemia, lack supply of oxygen, blood, so there will be feeling of pain. This pain is called ischemic pain. So it is the indication of coronary heart disease. And this is done, why it is done? It may be because of deposition of cholesterol in the inner lining of the wall. Okay, so there will be ischemia. There will be ischemia in the peripheral end. As a result, the cells will be hypoxic, and the nerve cell will not get energy, so it will lose its activity. It will be unable to send impulses to the center, or the center will be unable to control the distal part. Okay, so this is how, by creating pressure, we can injure a nerve. Another so this is ischemia, interference in blood supply. So what we are doing? Interfering with the blood supply. As a result, there will be less supply of oxygen to the nerve tissues, so it will be losing activity. When this is prolonged, then what will happen? There will be destruction of the suppose I have obstructed here, so there will be destruction of the myelin sheath, axis cylinder, axon, terminal part of the nerve. And this is, so you can see ischemia, interference of blood vessel can cause nerve injury. Another thing is transection, cut. If a nerve is cut, then it is called transection. So in that case, there will be injury. And crushing, in case of accidents, so there maybe the nerves can be crushed. So this is another kind of injury. These are severe. And injection of toxins and poisonous substances into the nerve bundle. So it will cause destruction of the nerve fiber. This is another kind of injury. And lastly, hyperthermia. If your body temperature rises above 106 degrees centigrade, then the nerves is going to die. Okay, so these are the several causes of nerve injury. So number one, transection, and number two, number three, number four, and last one. Okay. So now let us see. Now these are the causes. Try to remember all this. Now let us see, because of these causes of injury, the level of injury may be different. So accordingly, these are classified. Injury is classified. How injury is classified? Sunderland, a scientist, he classified the severity of injury, and according he classified as first degree injury, depending on the severity, is less to more severe. So this is less severe, mild, first degree injury. This is also called neuropraxia. So neuropraxia is A, B, C, D. Choose the correct one. Okay. So first degree injury is neuropraxia, and the second degree injury is axonotmesis. This is the also termed as we'll discuss in detail. And the third degree injury is Neurotmesis, then fourth degree injury, fifth degree injury. Let us see the type of this injury. First degree injury. It is most common type. Here, it is created by ischemia, obstruction in circulation. If the obstruction is for a little period of time, small period of time, then there will not be any structural injury in the nerve fiber beyond the level of pressure. So after some time, they will regain their activity. So there will not be any physical damage or morphological damage, structural damage. So that's why it is mild. So what is written there? It is caused by applying pressure over a nerve for a short period, leading to the occlusion of the blood flow and hypoxia. Okay, this is how, if you apply pressure, there will be hypoxia beyond this level. And action is not destroyed, but mild demyelination may occur. It will depend how long you are pressurizing the nerves. Axon loses the function temporarily for a short time, which is called conduction block. There is conduction block. The function returns within few hours to few weeks, depending on the pressure, how much pressure I apply. So this will depending on that, whether within hours or within days or within weeks, it will depend. Recovery. First degree injury is called neuropraxia, Seddon's neuropraxia. Let us see the second degree injury. So second degree injury is the prolonged and severe pressure. Here, the distal part, suppose I am to keep pressurizing here, so the distal part, there will be changes in the distal part. So, and we will see Wallerian degeneration. What are the degenerative processes? We will see the different degenerative processes. These degenerative processes will be seen here. Means there will be degeneration of the nerve. Physical means there will be morphological change, physical change, pathological change in the distal part of the injury. The endoneurium is intact. I have explained what is endonuria. What is endonuria? Again, I am coming back. This covering, endonuria. Okay? No. This is perineurium. Each fiber is covering. The covering of each fiber is endoneurium. Again, coming back. So this is endoneurium. Myelin sheath. Over the myelin sheath, there is another layer of sheath. So this is called endoneurium. for regeneration of nerve, whether it will die completely or it will be regenerated depends on this endoneurium. This endoneurium must remain. If it is destroyed, then there is no possibility of regeneration. So this is one of the precondition of regeneration. The endoneurium, here endoneurium is intact, so regeneration is possible. Repair and restoration of function take about 18 months. Second degree injury is called, what is this called? Axonotmesis. And third degree injury, it is called Neurotmesis. Third degree injury is called Neurotmesis. So you have to remember these terms. Nerve injury is specifically mentioned in your competencies. So you may expect questions in several examinations. The endoneurium is interrupted. So, endoneurium is deranged. It is interrupted. Epineurium and perineurium are intact. After degeneration, the recovery is slow and poor or incomplete. Why? Because of endoneurium is interrupted. Third, fourth and fifth degree of injury are called neurotmesis. So, this actually, the fourth also called neurotmesis, fifth degree injury is also called neurotmesis. Fourth degree, this type of injury is more severe. Epineurium and perineurium are also interrupted. In third degree, it was not interrupted. Now, these are also interrupted. Fasciculi of nerve fibers are disturbed and disorganized. So, everything is disorganized there. Regeneration is very poor or incomplete. And finally, the fifth degree block, it involves complete transection of the nerve trunk. The nerve trunk, it is complete transection of the trunk with loss of continuity. So there will not be any continuity. So cut, there is a cut, so arrangement is changed, and actually regeneration is possible if the both end lie close within 3 millimeter. If it is more than 3 millimeter, then it may not be possible. So this is another condition. The cut end, distal end, should be close to the, should remain close to the proximal end. So in case of severe cut, injury, accident, so what the doctor, surgeons do, if these are far away, they bring it closer by the surgery. So the regeneration comes possible. So let us see. So we learned about the causes of the injury. We learned the degree of injuries, level of injuries, first degree, second degree, third degree, fourth and fifth degree. And what happens in first degree? Why it happens? Second degree, what happens? Why it happens? Like this way, third, fourth and fifth. Okay. Now let us see what are the degenerative changes. One question comes, what do you mean by or explain Wallerian degeneration? Wallerian, Augustus Waller, In 1862, he spent his life only to study the degeneration of nerve fibers. He couldn't study regenerative process. He didn't know that actually regeneration can take place. At his time, he studied what happens to the nerve after it is injured and what are the changes he observed, and that changes, pathological changes or physical morphological changes, in functional changes in the nerve after injury, it was termed by his name is Wallerian Degeneration. So what is Wallerian degeneration? It is the pathological change that occurs in the distal cut end of the nerve fiber. That you understood, I told you repeatedly. It is named after the discoverer Waller. It is also called orthograde. The changes here will be orthograde, and there will be changes that proximal to the cell body. This is distal to the injury, and this is proximal to the cell body. So here also some degenerative changes can take place. Go. So that degenerative process is called retrograde. This is orthograde. Towards the periphery, orthograde. Towards the cell body, retrograde. Changes occur throughout the length of the distal part of the nerve fiber simultaneously. Not that this is the distal part. Not that here will be changes first, then gradually this will proceed, or it will be starting from here coming towards this. Not that. Simultaneously it will be happening. So what are these degenerative changes after disruption or cut or injury? So what happens in the axon? What happens in the cell body? Let us see. In the axon, what is there? Axis cylinder. Axon. This is axon, axolemma. Myelin sheath. Neurolemma. And again there will be a sheath. This is endoneurium. endoneurium. So what happens? Suppose cut is here. There are other fibers in the bundle in the fasciculi. So cut. Now let us see what is happening here. So first what will be there? This part will be swollen. This part will be swollen because of accumulation of water, accumulation of fluid. These Schwann cells will be disintegrated. They will die. They will be disintegrated. It will be converted into pieces. And this axis cylinder, the red part, this is a cylindrical structure now within this exoplasm is, so this is axis cylinder. Now this axis cylinder will be broken into various pieces. So what will happen? Myelin sheath will be broken and it will be disintegrated into small pieces. Axis cylinder will also be broken into small pieces. Okay? So what will be there? Only the endoneurium. Because we have to study this, how regeneration also takes place. For regeneration, we learned endoneurium is needed. So only endoneurium, in such cases, only endoneurium will be, if the injury is very severe, 3rd degree, 4th degree, 5th degree, then endoneurium will also be destroyed. So then there will not be possibility of regeneration. So here, in this case, in our case, for understanding, in this case, endoneurium is intact. Now, within the endoneurium, what has been there? It was destroyed. Now, there are some Schwann cells. These are made up of Schwann cells. These Schwann cells will also die. Since these Schwann cells are dying, their myelin sheath also will be dying. They will be disintegrated. This myelin sheath is made up of protein, mainly lipids and mainly proteins than protein. Mainly lipids. So they will be converted into lipids, droplets. Now, we know there are a lot of glial cells. We have studied glial cells. They are phagocytic in nature. So now they will come. They will enter here and they are macrophage in nature. So they will engulf all these debris. They will remove all these debris are within endoneurium. This is endoneurium, here is also endoneurium. Within endoneurium there are a lot of debris. Debris derived from the myelin sheath, debris derived from the Schwann cells, debris derived from the axis cylinder. So now all these debris are taken up by the macrophages and taken away. Now this area is cleaned so that regeneration can be take place. So these are the changes, and another change is there. This terminal is going to somewhere. Either it is making synaptic junction with another neuron. So this reaction will proceed up to that part, towards the end of this neuron, end of this section. And in severe cases, the next part also can show regenerative changes depending upon the severity. We have seen, if there is an injury in the cerebral cortex, so pyramidal tract, the nerves are coming from there. The injury is at the brain, where this pyramidal tract passing through the spinal cord, and in the spinal cord, different types of group of cells, we call them horn cells, H-O-R-N, horn cells. These are lateral horn cells, these are anterior horn cells, these are posterior horn cells. Anterior horn cells, posterior horn cells, lateral horn cells. They are actually connecting the nerves coming from the cerebral cortex, coming and ending in the horn cells. From the horn cells, new neuron. Horn cells are cell body. Horn cells are actually cell body. From this, their axon will rise, and this axon will pass to the periphery. Will pass to the periphery. Now see, if injury is in cerebral cortex, these are also affected because these are having continuity with the first order neuron. This is the second order neuron. First order originates from cerebral cortex, ends in spinal cord in the horn cells. Second order neuron, cell body is the second, these cells, and from them the second order neuron arises. So because of injury of the first order neuron, second order neuron can also die, undergo this thing. So this type of degeneration, it may continue in the next neurons also, beyond the synapse. Everything is discussed. See the image? Here is the cut injury. So this is the endoneuron. So see the endoneuron is intact and the axis cylinder is broken. Axis cylinder is broken into pieces. And see, these are the Schwann cell myelin sheath. These are breaking. Droplets of myelin. And what is the criteria? Yes, these are for regeneration, these are to be fulfilled. These criteria has to be fulfilled. Then only regeneration possible. What is this criteria? Number one, the gap between the cut ends of the nerve should not exceed 3 millimeter. Okay. Then neuronoma should be present. Second criteria. Thirdly, see, I have cut one nerve, one with sharp blade. The degeneration is very easy, a regeneration of this. If I cut a nerve with a blunt knife, then it will be, because it will be injury will be much more. This is the blunt, with the blunt one, I have to do it like this. And if it is sharp, one is enough. The cut end will be smooth. Cut end should remain in the same line. So this was the actual line. Now after cutting, the line became like this. Regeneration will not be there. Because this end, proximal, will produce something. Thinking that the axis cylinder is here. If axis cylinder is moved to this side, so this will not find the axis, something will be coming out. So these fibrils will be coming out. They are looking for the axis cylinder. But if the axis cylinder is moved there, they will not find the axis cylinder. So there will not be any regeneration. Okay. Journey time. 15 minutes relaxation time. Oh, where is that boy? Yesterday he was not attending. Who is that boy? Roll number 68 or something? He is not present today? You? No, not you, not this boy. Another boy, I said you go, others will attend the class. Who is that boy? Absent, no? Wow, very good. Because I will be continuing for another 10 minutes. So that's why he doesn't like my class also. Now see, there are 10 points. Stages of regeneration. Just follow one by one. First, some pseudopodia-like structures will be coming out from this part. They will produce some fibrils. From this, yes, some fibrils they will be producing some fibrils. Okay, these fibrils, they will proceed and they will look for, at the same time, here Schwann cells are died. Here the Schwann cells are not died. But in the periphery there are a lot of Schwann cells. They will be coming and they will multiplying. The Schwann cells will be multiplying, not within this. Previously one axis cylinder was there. Just outside the axis cylinder they will proceed. Multiply and proceed. Actually they will be guiding the fibrils that will be generated from here. They are guiding them, hey, follow this route. So they are creating a tubular structure so that the fibrils can enter within this. So here is the neurolemma. So outside the neurolemma, inside the neurolemma, these dividing cells are making a line. So within this, these fibrils will be entering. And from this fibril, new axillemma will be formed. Once new axillemma will be formed, from here exoplasm will be flowing. And once it is done, then myelin sheath will be formed. But in such case, only thing is 80% recovery may be possible, not 100%. The previous diameter was whatever it was, now it will regain 80% of the normal diameter. Okay, so this may take a lot of time. So here are these changes what I illustrated. So Golgi bodies, nasal granules, these were disappeared. Now everything, cell shape, that cell will lose water. Again, cell body will be regaining its original shape. And this endoplasmic reticulum, they will be forming new proteins, neurotransmitters, and nucleus will be shifted at the center. Okay, nasal granules will start to appear. So the way these were degenerated, now again these will be regenerated.